B12 then will hand off the methyl group, the carbon, to methionine. It goes through S-adenosine methionine and S-adenosine homocysteine and results in the formation of homocysteine. So when you see elevations of homocysteine, you may be worried about whether enough glutathione is being formed. If this cycle is going around on a constant basis and is active enough, then cysteine will be dropped off from homocysteine, which can then be used to form glutathione. So this is where we get the cysteine for glutathione formation. And this needs to happen in all our cells. And I want to give credit to Jill James for making us aware of this about 10 years ago in her publications in regard to finding defects in the MTHFR enzyme in children with autism. I have an interesting case that I'll go over with you in just a moment, in a youngster who had lowered glutathione levels but elevated taurine levels. And the preferential route is for cysteine to be used to make glutathione. But if it's not being used in the formation of glutathione, it can be used to form taurine. Here we have our GCL enzyme and our GS enzyme. And it's interesting to note that mercury chloride, for example, has been shown in vitro studies to inhibit the glutathione synthase enzyme. So here in review again, you have these pathways, the GCL and GS forming reduced glutathione. And I want to point out the role that G6PD plays in fueling the reaction to create glutathione reductase and reduce glutathione. If you don't have adequate G6PD, this reaction doesn't work as well. And the importance of these enzymes is that if you're not working in either one of these pathways, either the de novo production of glutathione or the recycling and reduction of glutathione, you can end up with problems. And this is because it's estimated that there's 50 hydroxyl radicals formed in each cell every second, or 4 million hydroxyl radicals per day in each cell. So this is a reaction that your body is using quite a bit, and explains why people who have deficiencies in G6PD may get into trouble. Now, the famous deficiency associated with G6PD is red blood cell fragility. It's been estimated, I've read, that 14 to 15% of the U.S. military has inefficiencies in this enzyme. This enzyme was associated with improving resistance against malaria, especially in northern African and eastern Mediterranean descent individuals. But when you give high doses of oxidizing agents, such as vitamin C, it's best to know that the person that you're treating does not have a G6PD deficiency. And here are all the vitamins that go along with supporting these biochemical reactions and these enzyme functions. And there's several I want to point out, such as the relationship of magnesium and B6 in the formation of and utilization of the GCL enzyme. Thank you. And so if you don't have magnesium, adequate magnesium, you'll become deficient in glutathione. And another strange factor that's not well known but is published is that if you're low glutathione, you may become low magnesium. So you need both magnesium and B6. And selenium is used the glutathione peroxidase is a selenoprotein. And there's some evidence that shows that raising selenium can raise glutathione peroxidase. You have to be careful a little bit with selenium, though. You don't want to raise it 1 to 200 micrograms. 1 to 200 micrograms per day is a good dose. You don't want to have an over excess of selenium because that can become toxic. At the same token, zinc is very important for the glutathione reductase and to keep that system working. You need all these enzymes working together to keep these systems functioning. I've had people suggest that you could just take all of these nutrient components and form glutathione, and I'm sure that that's true. However, when your system is stressed, we're going to show you some situations where these enzyme systems that perform the de novo restoration of glutathione may not be functioning. but it shows you the importance of these normal minerals in maintaining glutathione.